Yeah. Thank you. Good afternoon, everybody. My topic today is electrolytes and nephron. This is the crux of the acid-base balance and all the critical care headaches that we have. I won't delve into the actual acid-base disorders, but I'll give you the underlying physiology of the electrolytes that the renal tubules take care of. So the electrolytes in the body fluid compartments are the intracellular electrolytes and the extracellular. Among the intracellular, we all know the potassium is the highest, and among the extracellular, it's sodium. So the ECF and the ECF anions and the cations, that is the cations are the positively charged and the anions are the negatively charged. As I said, we have sodium, which is highest, and in the ECF anions, it is the chloride. So the ECF and the ECF anions and the cations, that is the cations are the positively charged and the anions are the negatively charged. As I said, we have sodium, which is highest, and in the ECF anions, it is the chloride. They maintain a state of electroneutrality, and among the intracellular fluid cations, it is potassium and phosphate. Now coming to the actual action of the renal tubules and why they are called the most intelligent part of the body is because of these intricacies of the electrolytes and the cations and the anions that they take care of. So first is the bicarb reabsorption, that is about 180 liters of glomerular filtrate is produced, and from that it is only 1 to 1.5 liters of urine that is produced per day. So the amount of fluid and the electrolytes that the renal tubule reabsorbs is the ideal, is in the center of these problems. So the bicarbonate reabsorption, 80% is absorbed in the proximal tubule, 15% in the thick ascending limb, and the rest 5% is in the cortical collecting duct and the medullary collecting duct. So in the proximal tubule reabsorption, what we have is 66% of the sodium, 85% of bicarb, 65% of potassium, which is mainly paracellular. All of the glucose and amino acids are absorbed. The transport is mainly paracellular and transcellular. The transcellular can be active or it can be passive. I would go very fast through my slides because of the time constraint. Schematic diagram shows you in the bracket A, that is in the diagram A, it shows the symporters, that is the SGLT2, the symporters, which is sodium and glucose, which are into the limelight nowadays. And there are others like co-transporters, sodium glucose, exchangers, sodium hydrogen, and chloride and base exchangers. Now this is a schematic representation of the proximal tubule. What is important is in the lumen we have H2CO3, which is broken down by carbonic anhydrase, and it forms carbon dioxide and water. Carbon dioxide enters into the cell. It is again combines with water to form carbonic acid, and there the bicarb is again absorbed through the basolateral membrane. And if there is a defect in this proximal tubule, be it ischemic injury, be it genetics, then it results in mayhem. That is, all the bicarb is not reabsorbed and other electrolytes are also down into the urine. Bicarb reclamation, as I said, is mainly through the NHE3 transporter. The vacuolar H plus ATPase is also involved. And when we have the proximal RTAs, that is the renal tubular acidosis, it is this the basolateral cation exchangers. Now this is about the net acid excretion. The important part is mainly determined by ammonium. Ammonium here is glutamine is broken down into ammonia, which combines with hydrogen ion, and it is exchanged through the NHE3, and it forms ammonium in the luminal part of the proximal tubule. So the cytosolic ammonium can diffuse into the lumen to trap the, I'm coming to the third line directly, and ammonium then passes into the medullary thick ascending limb. This is the medullary thick ascending limb. Here is another important part because this importance lies in the Barter Syndrome, which we are all familiar with. We know the loop diuretics blocks the NKCC2 receptors, which is labeled as 1. and the deficiency of this results in Barter 1. Next, we have the ROMK channels, that is the apical potassium channels, which is involved in Barter 2. The CLC-KB, which is labeled as 3, is the Barter type 3, and Bartine associated with it is the type Barter 4, which results in Barter syndrome along with sensorineural deafness. Now coming to the I'll go very fast. Another very important is the calcium sensing receptor, which is present in most of the tubules. But here what happens is activation of calcium of the basolateral calcium sensing receptor inhibits sodium transport by inhibition of NKCC and ROMK, that is the NKCC2 and the ROMK, thereby decreasing the transtubular potential gradient, driving the calcium and magnesium reabsorption. Calcium and magnesium is mainly paracellular transport through claudin 16 and 19, and loss of function mutations of these claudin 16 and 19 proteins will result in hypercalciuria and hypomagnesemia. In the distal tubule, as we all know, it's the sodium chloride receptor which is important, marked as 1. And these are blocked by the thiazide diuretics. Now these are influenced by the WN kinases 1 and 4. If there is an activating mutation, what we have is the pseudo-hypoaldosteronism type 2. These are all actually involved in, means our MCQ knowledge. We are not bothered with the actual patients. The Gitelman syndrome, of course, we get a lot of patients which result in inactivating mutations of the sodium chloride channel. In the late distal tubule and the cortical collecting duct, we have two types of cells, that is the principal cells and the intercalated cells. Here in figure A, we'll see the principal cell. The point of importance is the epithelial sodium channel. This is labeled as 1. The epithelial sodium channel causes sodium absorption, but it is under the control of aldosterone hormone. And we have mutations of epithelial sodium channel resulting in Liddle syndrome, that is the activating mutation. If we have an inactivating mutations, it will be pseudo-hypoaldosteronism type 1, resulting in salt wasting. And in the type B, in the figure B, what we find is the water absorption, that is the pathophysiology of nephrogenic diabetes insipidus. It is the V2 receptor, which is under the influence of arginine vasopressin, as we can see is labeled as 2. If that is defective, it results in the X-linked nephrogenic DI, and if the aquaporin channels are not put into the luminal membrane, then it results in the autosomal recessive nephrogenic DI. In the left lower quarter, we'll find the pendrin channels, that is the chloride transporters and the sodium and bicarbonate transporters. Here, if pendrin is defective, we know Pendred syndrome. And on the right hand side, we find the A type of intercalated cell, which result in proton secretion into the lumen. So this I was talking about. This is nothing new. This is another representation of type A and type B intercalated cell. And in the medullary collecting duct, as we all know, the water absorption is ADH dependent, that is AVP dependent, whereas in the thin descending limb, it is ADH independent through the aquaporin 1. So this is a recap. This is the proximal tubule, sodium chloride, bicarb, potassium, water, glucose, amino acid, everything goes in. And what we have is hydrogen, organic acids, and bases. This is the thin descending limb of the loop of Henle. Water is reabsorbed through aquaporin 1, ADH independent. In the lower figure, it is sodium chloride, potassium, calcium, magnesium, and bicarb in the thick ascending, medullary thick ascending limb. Here the fluid is hypoosmotic. And the early distal tubule, again sodium chloride through the thiazide sensitive transporters along with calcium, magnesium. And in the late distal tubule, it is again sodium chloride. Whatever was left is mopped up with the help of antidiuretic hormone. And this is the medullary collecting duct. So ultimately, the net bicarb excretion is 0. And here, these are the list of the entire gamut of genetic mutations that can occur. And that's, thank you. I think I finished in time.